keeping up with the sheer volume of scientific knowledge being generated today feels, well, almost impossible, doesn't it?
Oh, absolutely. Every hour, new papers, new data.
It's just a constant stream, a fire hose, really. And trying to synthesize all that, let alone find genuinely new connections hidden inside, that seems like a huge task for anyone.
It is. It's a monumental challenge. And you sent over a really interesting set of sources looking right at this problem.
Yeah, especially how it impacts things like drug discovery.
Exactly. That's where you really see this lag that the sources talk about. You have valuable insights, maybe about a new use for an old drug, just sitting in the literature for years.
Sometimes decades, right, before someone finally connects the dots.
Precisely. The sources mentioned dabrafenib, the cancer drug. Its effects on hearing were known, but using it for hearing protection took ages.
And ketamine, too. A 22-year gap for its antidepressant use. That's striking.
It really is. Leucovorin, CarXT, the list goes on. It shows we need better tools.
Tools that can, what, read faster? Or is it more about making connections?
Both, really. Processing information faster helps. But the key is making those non-obvious connections. Linking findings across maybe totally different fields.
Right. Which brings us to the mission for this deep dive, looking at your sources on Robin. This new AI system that's designed, apparently, to tackle this exact problem, accelerate discovery.
Yes, and what makes Robin stand out, according to these papers, is its ambition.
How so?
Well, it's not just another AI tool for, say, literature search or data analysis. The claim is it's the first system aiming to automate the entire core intellectual cycle.
The whole loop, from idea to results and back again.
That's the core innovation they're pushing, going beyond automating isolated steps to connect everything. Background research, hypothesis generation, proposing experiments, interpreting the results, and then generating updated hypotheses, all in one continuous workflow.
Okay, that sounds ambitious is the right word. So let's unpack that. How does it actually work? What is Robin? The sources call it a multi-agent AI system.
Yeah, and that multi-agent part is really key to understanding its structure.
Why is that significant?
Because it's not like one single AI trying to be an expert in everything. Think of it more like a specialized team.
Okay.
Robin is kind of the coordinator, managing these different AI agents, each designed for specific complex tasks.
Like different specialists on a research team.
Exactly like that.
Yeah.
It makes the whole system more modular, efficient. You can improve or swap out one agent without rebuilding the whole thing.
Got it. So what are these key intellectual steps that these agents handle? You said the whole cycle.
Right. So it mirrors how scientists work, essentially. Gathering background info, like literature reviews, formulating very specific testable hypotheses.
Okay.
Then, crucially, proposing concrete experiments like suggesting specific assays or cell models to actually test those hypotheses.
So it suggests actual lab work.
Yes. And then when the data comes back from the lab, it analyzes those results, often complex data.
And interprets it, figures out what it means for the hypothesis.
Exactly. And then uses those insights to refine the hypothesis or generate new ones for the next round. It closes the loop.
So it's not just pattern finding. It's proposing actions, analyzing outcomes, and learning iteratively.
Precisely. Integrating all those stages is what the sources highlight as the big step forward. It operates in this sort of lab-in-the-loop framework.
Lab-in-the-loop, meaning it works with human scientists.
Yes,
Yeah.
semi-autonomously. The humans do the physical experiments Robin proposes, get the raw data, and feed that data back into the system.
Okay, semi-autonomous. The humans are still critical for the wet lab part. Makes sense.
Absolutely.
So who are these specialized agents, this AI team? The sources mentioned 3 names, Crow, Falcon, and Finch.
That's right. Each has a specific job. Crow is the literature expert.
Okay.
It does rapid, targeted lit reviews, finds relevant papers, identifies potential experimental strategies, even pinpoints candidate drugs or molecules to test. Think of it as the super librarian and strategist.
Finds the leads, basically.
Exactly. Then there's Falcon. Falcon takes the candidates Crow finds and does a deep dive.
A deeper dive how?
It generates detailed evaluation reports, looks at the scientific rationale, why a candidate might work, but also flags potential limitations or problems, really assesses the potential.
Okay, so Crow finds, Falcon evaluates, and Finch.
Finch is the data analyst. It's designed to autonomously analyze complex experimental data. Things like flow cytometry output or RNA sequencing data, really sophisticated stuff.
So Robin coordinates Crow to find strategies and candidates, Falcon to vet them, and then Finch to analyze the results when they're tested.
You got it. That modular structure lets it tackle these big, complex problems piece by piece.
It sounds very logical, actually, like breaking down a massive research project.
Yeah, it reflects that kind of collaborative expertise you'd see in a big research group.
Let's walk through that workflow then. Using the example from the sources, dry age-related macular degeneration, DAMD, how does Robin start on something like that?
Okay, so it starts with the human input. The scientist gives it the target, DAMD.
And DMD is a big deal, right? Major cause of sight loss, not many treatments.
Exactly. Huge unmet need. So Robin gets DMD. First, it uses Crow to scan the literature and generate potential disease mechanisms, like what could be going wrong biologically in DMD. In the case study, it came up with, I think, 10 initial mechanisms. For each one, Crow then proposes specific in vitro models, lab setups, essentially, and assays to study that mechanism.
Right, ways to test ideas in the lab. And this is where another innovative piece comes in the LLM judge. What's that?
Yes, that's a really neat technical bit. Robin uses a large language model, an LLM, specifically trained to act as a judge.
A judge of what?
Of the experimental strategies Crow proposes. It ranks them. It does this through pairwise comparisons, basically asking the LLM, okay, here are strategy A and strategy B. Which one looks more promising? It repeats this until it has a ranked list.
Huh. Using an LLM to rank scientific strategies, does that work?
Surprisingly well, according to the sources. They found its rankings had high concordance with human experts. It matched something like 7 out of their top 10 preferred hypotheses. And interestingly, the LLM judge was actually more consistent in its own repeated rankings than the human experts were, like 88% consistency versus 61% for the humans.
That suggests it's a pretty robust way to automate that selection process.
It seems to be. It helps Robin zero in on the most promising experimental path. So for DMD, the chosen strategy focused on enhancing phagocytosis in retinal pigment epithelium cells, RPE cells.
RPE cells. Quick refresher, those are key support cells in the retina, and phagocytosis is their cleanup job, right? Getting rid of debris.
Exactly. And if that job fails, debris builds up, which is a hallmark of AMD. So enhancing that cleanup seemed like a good therapeutic strategy.
Okay. Strategy chosen. What next?
Robin directs Crow again, this time for a more focused literature review just on RPE phagocytosis. Crow reviewed about 400 papers and proposed specific therapeutic candidates, drugs, or molecules that might boost this process.
And then Falcon steps in.
Yep, Falcon generates those detailed reports on Crow's candidates, outlining the why and the potential buts.
And then another judging round.
Another LLM judge tournament, yes, to rank these drug candidates based on Falcon's reports and the literature. This gives the human researchers a prioritized list of what to actually test in the lab.
Okay, so the humans take that list, run the experiments, let's say using that RPE phagocytosis assay, gather the raw data and feed it back to Robin.
Correct. And now it's Finch's turn.
The data analyst agent.
Right. You feed Finch the raw data flow cytometry data in this first round for DMD, measuring how much the cells were eating.
And Finch analyzes this autonomously. What's special about how Finch works? The sources seem to highlight this part.
Yes, it's autonomous, and this is a key technical innovation. Biological data interpretation isn't always straightforward, right? There might be multiple valid ways to analyze it.
Sure.
And LLMs themselves can have some randomness, some stochasticity. So Finch doesn't just run the analysis once. It launches multiple analysis trajectories.
Multiple trajectories? What does that mean?
It means it explores the data using several different valid analytical approaches at the same time. For complex stuff like RNA sequencing, it might run, say, 10 different analyses in parallel.
Okay.
To ensure robustness. It then synthesizes the results from all those trajectories into a consensus conclusion. If multiple different analysis paths point to the same result, you can be much more confident it's real and not just an artifact of one specific method or a random fluctuation in the AI.
I see. That's clever. It's like getting a second, third, 10th opinion on the data automatically.
Exactly. It builds in robustness against the variability. And Finch does this within a structured environment, like a Jupyter notebook, using frameworks like Aviary and specific prompting techniques like React, so the process is transparent and reproducible.
Okay, so Finch delivers its robust, synthesized analysis. Then what?
Then Robin takes Finch's analysis and interprets it in the context of the original hypothesis. Did the experiment support it? Refute it? Did something unexpected pop up?
And uses that to update the plan.
Yes. It refines the hypotheses. Maybe confirms one, maybe suggests modifying another, maybe even sparks a completely new line of inquiry based on the data. It closes that loop, feeding the results back into the knowledge base to guide the next cycle.
Got it. So let's really nail this down with the DAMD case study. Show us the aha moment. Robin started with the hypothesis. Boosting RPE phagocytosis could treat DAMD.
Right. Based on the literature suggesting RPE cleanup fails in AMD, so proposed candidates.
The researchers tested them using Frodo beads. Those light up when engulfed.
Yeah, exactly. A flow cytometry assay to measure phagocytosis. Finch analyzed that data from the first round. What came out?
Finch confirmed that one candidate, Y27632, which is a known ROCK inhibitor drug, did significantly boost RPE phagocytosis. Okay, so validation. The system's reasoning based on the literature held up experimentally. That's good.
It is, but like you said, maybe not revolutionary on its own, but here's where, as the sources put it, it got really interesting.
Yeah, what happened?
Robin didn't just say, okay, Y27632 works, hypothesis confirmed. It recommended a follow-up experiment.
Based on that result.
Yes. It suggested doing RNA sequencing on the RPE cells that had been treated with Y27632.
Why? To understand how it was working.
Precisely. To get at the mechanism. This shows Robin driving towards deeper biological understanding, not just observing an effect. Finch then performed that complex RNA-seq analysis, differential gene expression, pathway analysis, the works.
And found something unexpected.
Totally unexpected. The analysis revealed that Y27632 treatment rapidly and significantly upregulated a gene called ABCA1.
ABCA1, what does that do?
It's a critical protein involved in lipid efflux, basically, pumping lipids out of cells.
Okay. And why is that significant for DMD and this drug?
Because while Y27632 is known to work by inhibiting RCK, affecting the cell structure, this ABCA1 finding suggested a whole other mechanism might be at play. It implied Y27632 might also be improving RPE function by influencing lipid metabolism via ABCA1.
Ah, so it wasn't just the known pathway. It pointed to a potentially novel target or effect.
Exactly. It connected ROCK inhibition, phagocytosis enhancement, and lipid handling in RPE cells in a new way, directly linked to this DMD context. A genuinely new insight generated from the experimental data analysis by Finch.
That's a fantastic example. The AI didn't just confirm, it used the data to generate a novel, testable hypothesis about the underlying biology.
Right. And that new insight, that ADCA1 connection, fed straight back into Robin's next cycle.
So it used that refined understanding to propose new candidates.
Yes, candidates that might leverage this pathway or be even more effective. The researchers ran a second round of experiments with these new suggestions. Finch analyzed that data.
And the winner was?
Ripasudil. Finch's analysis showed ripasudil boosted RPE phagocytosis even more strongly than Y27632 in our assay, something like a 7.5-fold increase.
Ripasudil. I feel like I've heard that name in relation to eye treatments.
You probably have. It's also a clinically approved ROCK inhibitor. But, and this is the kicker, it's approved in Japan for treating glaucoma.
Glaucoma, not AMD.
Not AMD. According to the sources, despite being an eye drug and a ROCK inhibitor, ripasudil had never been proposed for DMD before Robin identified it through this process.
Wow. So Robin's whole iterative loop, literature, hypothesis, experiment design, deep data analysis, leading to new mechanistic insights, refining hypotheses directly pointed to an existing approved eye drug as a potential treatment for a completely different eye disease.
Exactly. It found a promising repurposing candidate, one with an existing safety profile for eye use. That's a direct hit on accelerating discovery and overcoming that lag.
That really demonstrates the potential power here. Finding a shortcut like that could save years and millions in development.
Potentially, yes. That's the hope.
So zooming out from DMD, what are the bigger implications here, if this works?
Well, the potential is huge.
Yeah.
The sources mentioned Robin generated hypotheses for 10 other diseases too. It could, in theory, dramatically speed up discovery across biology and medicine.
By doing what exactly? Synthesizing information better, proposing smarter experiments.
All of the above, synthesizing vast amounts of data efficiently, proposing novel or non-obvious experiments based on that synthesis, and then extracting deeper, more robust insights from the resulting complex data, like Finch did with the RNA-seq.
It sounds almost too good to be true. What are the limitations? The sources must mention some downsides or challenges.
Well, for sure. They're quite open about it. It's not magic yet. For one, Robin proposes experimental strategies and ranks candidates, but it doesn't spit out, like, a precise step-by-step lab protocol ready to run.
Ah, okay. So human scientists still need to translate its ideas into practical lab work.
Definitely. Human expertise is still crucial there. Also, Finch's data analysis, while powerful with the multi-trajectory approach, still needs some expert guidance and prompt engineering, especially when faced with totally new types of data. Making it even more autonomous there is future work.
Makes sense.
And finally, they mentioned continuing to refine how the LLM judge evaluates hypotheses to better capture all the nuances of expert human scientific judgment. There's always room for better alignment.
Okay, so known limitations, areas for improvement. But they sound more like engineering challenges than fundamental roadblocks.
That seems to be the perspective, yes. The core achievement, they argue, is demonstrating that automating the full intellectual loop is feasible.
Establishing that new paradigm for AI in science.
Exactly. Moving AI from just being a tool for one step to being a semi-autonomous partner driving the whole discovery cycle alongside humans.
So if we boil it down, the big takeaway from these sources is Robin represents perhaps the first real attempt to fully automate that core intellectual engine of science, digging through knowledge, forming ideas, testing them, learning from results, and repeating.
That's the central claim. And the potential, as shown with ripasudil, is accelerating therapies, finding those hidden connections for drug repurposing, and ultimately driving a much deeper understanding of biology by testing ideas faster and more systematically than we ever could before.
It really makes you think. Considering Robin's knack for connecting disparate info and pulling out unexpected insights like ABCA1 from that Y27632 data, you have to wonder what other big, complex scientific puzzles where maybe all the pieces are already out there in the literature but just disconnected could a system like this start to put together?